Good morning, everyone. It's my pleasure to welcome you as well to our conference call this morning on our top-line phase 2A data. We're thrilled to share these data with you, and we're very happy that we got to this point. So we will dive right in. Before we do so, just two notes here. I will be joined by our Chief Medical Officer, Dr. Camilla Chong. Dr. Chong will present the data in chronic spontaneous urticaria, and I will present the data on Hidradenitis suppurativa. We're also thrilled to have with us for the Q&A session Professor John Ingram. Professor Ingram is not just a treating physician that is passionate about HS treatments and knows a lot about that, but he's also one of the key opinion leaders and really key scientists driving the field forward in HS. As a former editor-in-chief of the British Journal of Dermatology, also very well versed in the scene, and we're really thrilled to have him with us for the Q&A session. So with that, we'll dive right into the presentation. So please, next slide. So we'll be making forward-looking statements today. Next slide, please. And please take note of the disclaimers. These are important disclaimers on the forward-looking statements. We will also be showing you for comparative illustration only some data that were presented by and reported by other companies. So this is for illustrating purposes and comparative analysis only. Next slide, please. So just on the mechanism, as a reminder, we have a small oral compound inhibitor of the C5a receptor, sometimes referred to as C5aR1. The C5a receptor is a known and validated target in an immunoinflammation field, and obviously it's part of the complement system. And a small inhibitor of this receptor has certain advantages. It has a deep tissue penetration potential, which acts then in the site of where the inflammation really takes place, in this case, the skin. And also, it's not really dependent on how much C5a is being produced. With that in mind, it's important to know that you cannot simply block the C5a receptor with upstream inhibitors because C5a can be produced by various pathways, particularly through enzymatic cleavage on the cellular level in the skin, but also in different tissues and also in the blood. So to block this pathway, you need a targeted inhibitor. Next slide, please. We have previously shown data, and this is from our phase 1 study here, that indicated that INF904 has best-in-class potential. And this is really based on a very differentiated PK profile next to the reported comparator, which is a marketed drug, avacopan. And you see the data here as a reminder on the upper right corner. So at the same dose already, we reach a 10-fold area under the curve increase and a very differentiated ability to block the receptor and really have a tighter control over the signal. Why is this important? Because ultimately we believe, and some of the data today shown will hopefully make that point again, that with that we can explore efficacy to a much higher extent, and eventually this will lead to higher efficacy signals in clinical studies and in clinical application. I just also want to remind you that by now we have, including the data we will present today, about 180 human subjects exposed to the drug with no signals of safety concern. Next slide, please. So this was a PK and safety study. So we will start with PK and safety. PK at this point is very preliminary. You see that on the right side, we plotted a compiled PK data in green HS and in blue from the CSU study. But the illustration here is not complete. You see the number of subjects are indicated underneath the figure. The point we're trying to make is it's very differentiated from the marketed comparator. Already at week 1, we have a multiple higher exposure for both indications across the 3 tested doses in HS and the 2 tested doses in CSU. And even if you see the plotted line here from avacopan from its approval data in ANCA vasculitis reaching at 13 weeks steady state, even if you compare that, we clearly exceed that sometimes by a multiple. And so we just wanted to also let you know we started with 60 mg BID, and that was based on the knowledge that 60 mg BID is actually covering the signal quite well. In our phase 1 studies, we've been able to demonstrate that. So we believe 60 mg is a clearly active dose, but we wanted to explore efficacy upwards, which in skin diseases is particularly important as oftentimes when first tested, drugs are underdosed in this. Next slide, please. So safety. Very high level, you find more details in the appendix with the concrete listings. There are no signals of safety concern detected in either of the arms. There's no reported SAEs in either of the arms, so no severe adverse events. And there is just 3 adverse events of mild grade 1 nature reported in 2 patients in the HS cohort, and 1 particularly also mild grade in the CSU cohort. So very, very clean safety profile so far, and we're really happy to report that we have no signals of safety concern. Next slide, please. So with that, we jump into the HS dataset, and I'm thrilled to be able to be presenting this for you today. We have 29 patients as depicted here below that we will be showing you today that have reached the end of treatment at week 4. And out of these 29, 25 have already reached the end of study, which is week 8. So 4 weeks later without dosing and without additional visits. The other 4 remain and have not reached that endpoint yet, but are in the study. Please also take note there's 1 patient in the low dose group 60 mg and 1 patient in the medium group 90 mg that is still finishing up treatment and data entry, so they are not included in this. The high dose group therefore is complete, but the other 2 dose groups each missing 1 patient that are still in the treatment phase. Next slide, please. So just as an indicator, again, this is the setup. The color coding will be consistent throughout the deck. So yellow will be the 60 mg BID group, pink is the 90 mg, and red the 120 mg. You see here the 4-week dosing intervals. Again, dosing is every day BID with 4 visits after the baseline visit. And then another 4 weeks, as I mentioned, no dosing and no visits, where the patients are just coming back 1 more time for a safety and efficacy analysis at so-called end of study week 8. Next slide, please. So with that, we are showing you here the baseline of the 3 groups, 60, 90, and 120 mg, and below that is the combined data of all 3 dose groups. You will see that they're overall pretty balanced and in line with previously reported and conducted HS studies, maybe highlighting that on the AN count, which drives an important regulatory endpoint, the so-called high score, the N count is the highest in the 120 milligrams dose group and the lowest in the low dose group. There's a difference that is noticeable. The DT count is between 3.5 roughly and 4.5, in line with other studies. Also, the severity score indicates that the high dose group is the most severe, with the so-called IHS4 score. And then maybe also noteworthy that we have a few prior biologic experienced patients and a slightly higher BMI, body mass index, on the upper right in the low dose group. Next slide, please. So we will now show you the lesions first and then the scores, and we start with the driver of the high score, the so-called AN count. So just as a reminder, abscesses and nodules are 2 of the inflammatory lesions. The third one is the draining tunnel. Abscesses and nodules come and go all the time, so they fluctuate a lot in the patients. So they can go up and down by over 50% in lower count patients, and that's very normal. You see on the upper left side here how the average in each dose group is composed with respect to the nodules and the abscesses, making the AN count altogether. And as you can see, the high dose group is the most severe one. I should mention that in green, we always depict the compiled data of all 3 dose groups. So when looking at that, we would like you to appreciate first that you see a very consistent overall reduction. And this is week by week data, so high granularity in the absolute reduction of these counts. Now you see that, oftentimes there's a lot of zigzagging, there's flaring patients. So just 1 flaring patient can cause a lot of zigzagging in this curve. So we were very happy to see a very consistent move. And you can see this is exceeding 8 total abscess and nodule counts already at week 4 in the high dose group. So why is this important? How does it comp? Next slide, please. We have done a lot of comparator analysis that we will share with you, in which we compiled the data that have been reported for successful drugs and created the average of that. And you will always find in blue completed and successfully conducted phase 3 data sets that are averaged, and in gray phase 2. In this case, for example, it was just 1 drug in phase 2, povorcitinib, and we then report the best reported dose. And in phase 3, of course, the doses that have eventually then also led to an approval. And you can see the corresponding placebos are always in the dotted lines. So blue is phase 3, gray is phase 2. So you see that the green line of all doses here compiled is just on par with the reported successful drugs here. but the red line is clearly exceeding this. And the other thing that we can learn from this orientational comparison is that the placebo ability to reduce the AN count is across the different studies that we looked at, always in the range of -2.5 and -3.5. Now that is not unimportant because they drive the high score. As a reminder, the key criterion is a 50% reduction of the AN count from baseline. So if you have a baseline of 6 counts, you need to lose, for example, 3 nodules, and you reach that criterion, 50% reduction. But if you're coming from 30, you'd have to go down to 15. And if you see that the average placebo makes about 2.5 to 3.5 points, you can already kind of see from that this is oftentimes driven and drives response in the low count patients. So the low count patients provide a lot of noise, and they can drive placebo response rates in that score. Next slide, please. So looking at draining tunnels, we had mentioned previously and guided the markets that draining tunnels are an interesting lesion, as we can see them as an indicator lesion. Why? Because historically, they're very low response rates for placebo trials. So the draining tunnels are more consistent, less fluctuating. One draining tunnel can produce up to 1 liter pus per day. Just want to let you know that a draining tunnel is what you see to the outside, but this usually means if you, for example, drain large milliliters of up to 1 liter of pus from one draining tunnel, that there's a big underlying large inflammatory lesion in the deep skin that produces an inflammatory lesion that produces this pus. And so reducing draining tunnels is a remaining unmet medical need, but it's not easy, and it's usually not done by placebo responses. So here you see the same picture, different doses, again, the high dose performs the best. And on the right side, you see something that was created recently by calling DT100, meaning how many patients that have draining tunnels at baseline are now completely draining tunnel free. I just want to remind everyone, we're talking about 4 weeks of treatment. So at 4 weeks in the high dose, we have half of the patients already draining free, and the average around 30%. Next slide, please. How does it compare? Pretty impressive in my eyes. As you can see, the placebo from the phase 3 trials, by the way, I should have mentioned, always underneath you find all the trials, all the data that went into the curves. In this case, it's adalimumab, secukinumab, bimekizumab, povorcitinib. So everything that was recently reported here is in there. And you see placebo has very little reduction of the draining tunnels. And you see that the compiled successful drug data here, they reach about a bit over 1.3 point reductions overall. And you see, no matter whether you report on all patients or only on those that have draining tunnels at baseline, which in this case makes the difference between dotted solid red and green, there's a very steep initial decrease that we can note here for our drug INF904. So next slide, please. So how does that funnel into the high score? We now looked at the lesions. The lesions are part of high score. I should mention reduction of draining tunnels is not rewarded and not looked at in the high score. You should just not have increases. So as we explained, there's a lot of noise in the high score. There's ups and downs, and usually you don't get granular weekly data as we can show here. But again, we're very happy to report that this is moving upwards step by step, with reaching about 38% in the higher dose group already at week 4. And then remarkably, when we took the patients off drug, those 25 that reached the end of the study, the additional 4 weeks off drug, the responses deepened. Now, obviously, the denominator is just 25, not 29 here, but the high dose group is complete. So that 63% you see there is not changing anymore. The other 2 groups have still a few patients coming in, so they could still change to a higher or a little bit lower level. But the point is that response is deepening. And we, of course, looked into how can that be explained. First of all, we can confirm that the preliminary PK data at the end of the study, so 4 weeks off drug, do still show active drug levels in the range above the IC50 of the drug. So where there can be a blocking ability still from the plasma levels being estimated. So that's one thing we want to note, but also the mechanism does provide for a change in the inflammatory environment in the skin, meaning you will have less of certain immune cells moving into the skin over time and being activated in the skin, and that can also have a carry forward effect. On the right side, you see something that we created to illustrate how high score moves overall. And so for this, it's the pooled data. And we said, knowing about the variability in small data sets, how can we depict and create confidence that there is a movement towards a higher high-score rate. And the way we did that is we created this stacked bars here. We're looking at high score 30, then high score 40, high score 50, and high score 75. And we said, look, if without the, with knowing about the variability over time, these stacked bars should get higher as more patients move into the next response level. And obviously, if you're at high score 40, maybe you need to lose just one nodule or one abscess, and you're already in high score 50. And this is exactly what we see here. These stacked bars, they extend, they get larger, and we're at a high score 75, then at 24% at week 8, which again is something that we wanted to depict. So next slide shows you then how that compares. So again, depicting the phase 3 studies. First of all, you see visually that placebo is not so much differentiated, and we know about that problem. Very recently, we got reminded again that placebo response rates do play a role. I want to mention again that can be mitigated when looking at the baselines in different ways and how you control not to have too many low AN count patients in your placebo group. But we are right on the blue trajectory line, obviously with some variability and obviously with the high dose again performing better. But I would say clearly we are in the range that we can check the box in high score. And have the fantasy that may even move higher over time, and that needs to be proven out in future studies. Next slide, please. So the last remarkable point here in the HS data set is really the pain scores. NRS 30 meaning on the pain scale from 0 to 10, you get a ground score. You will notice in the baseline that this was roughly between 6 and 7. So these patients are highly impacted by pretty intense pains. And it's rare that you have a consistency in small data sets because this, individual score, which is a patient-reported outcome, has a lot of variability. But you see a consistent improvement alongside the lesions improvements that we've been showing you in the pain up to a pretty remarkable level in average over 65% and 75% in the high dose group. Now that is important. We defined high score NRS 30 as having at least a 30% reduction from your base score and at least a 2-point reduction, which is a bit more stringent than some others used. A recent competitor read out a different way by saying we just want to show you patients that have at least a 3-point reduction. So the NRS 30 was not part of that, but we also applied that logic and the lines did not change by chance. So no matter how you look at the data, they're really in the comp that it's shown, they really kind of stand out in as much as they're very consistent and they're showing a very, very large improvement in very early in the first 4 weeks. And that's maybe in line again with treating the inflammation in the skin. Next slide, please. So this is less of a efficacy demonstration, it's like more of a comparator slide. We mentioned that we think we have a best-in-class potential drug that is shown and supported by our phase 1 data. And I think this supports it again because avacopan was used in HS in a pretty sizable study, roughly 120 patients per arm. And you see this is the data that are reported on clinicaltrials.gov about this study. And you see that for DT, draining tunnel, this was plotted out over the time phase of 12 weeks. And you see how we compare next to their approved dose 30 mg, which did not really separate from placebo. And on the right side, you see a bit of an apple and pear comparison, our data at week 4 and their data at week 12. And when looking at AN count that drives high score, you see that there is a signal in avacopan, maybe even a dose response. Again, you see that placebo is within the range I mentioned to all of you between 2.5 and 3.5. But there is a signal in avacopan, maybe not as strong as a signal as bimekizumab can do, maybe up to 7 or so at that time point, or even over 7. But it's just reaching this very late, and it's not very impressive. But we have reached this already in average at week 4 and clearly exceeding with our best reported dose at week 4. Similarly, the pain scores that I already illustrated, there's a clear difference. So we believe this is another good indicator that the drug is very differentiated and has activity away from the marketed comparator. Next slide, please. So with that, we conclude here. So we believe that this data here show a biologic-like efficacy in the first 4 weeks with the N counts and high score responses. We see the high score responses deepening 4 weeks off drug, and we have an explanation for that, and that's a very encouraging signal. We see fast and deep reductions in DT, and that really is very differentiated from reported placebo and also compares favorably to the phase 3 drugs. And also we showed you a very consistent reduction in pain In the backup, you will also find data on dermatology life quality index, which are equally impressively improved already at week 4. So that's in line with the lesion reductions. And I mentioned safety, that we don't have a safety signal of concern. And with that, we think we deliver to the market here a very interesting, a very differentiated mechanism of action that is an oral, and we're very excited to take this drug further in this indication. With that, I'm done with my part. I'm happy to hand over now for the CSU part, which also shows clearly very interesting signals and an active drug. And I would like to hand over to my colleague, Camilla. Would you be so kind and take it from here? Thank you.
Thank you, Niels. I'd be delighted to take the CSU part of our presentation. But before I present results from our CSU study, I would like to take the opportunity to acknowledge the late Professor Marcus Maurer from Charité Berlin, who really persuaded and collaborated with us in this to embark on this CSU study, because he really believed in the crucial role that C5a plays on not just the mast cell, but also the underlying inflammation. And we are grateful that his hospital continued to work with us and is in fact one of the sites in this particular study. So for the purpose of today, I will be presenting 30 evaluable patients who have completed treatment up to week 4. And out of those, we have 23 right now who are have data at end of study. The remaining 7 are still ongoing, and we also have an ongoing 3. Next slide, please. As Niels have shown you, the study design is fairly similar, except for CSU. We do not have the 90 milligrams group. We have the 60 and 120 milligrams BID. We also have arm 3, of which we try to enrich the 120 milligrams dose group with patients who have previously been treated with anti-IgE and who have either had incomplete or no response, and also those who present with low IgE, more indicative of the type 2B endotype. These patients have 4 weeks of treatment, and then from end of treatment to end of study, no drugs are dosed. So basically, they have a dosing free period, but we follow them up. With that in mind, I'd like to show you in the next slide the UAS, first of all, baseline characteristics. So as you can see, the mean age of these patients are around 44 years old, predominantly female, with quite severe, moderate to severe CSU at baseline, and also poorly controlled disease. On average, they have had CSU for at least 3.5 years. We have a group of patients who are anti-IgE experienced, but predominantly anti-IgE naive. In the next slide, please, I will share with you the UAS7 results at week 4 of both doses, 60 milligrams, achieving a UAS7 reduction of just under -14, and the 120 milligrams group at -8. On average, we achieve an end of treatment results of around -10.4. And I've mentioned earlier to you, from week 4 to end of study treatment, where they have no drug being dosed, the effect on the 60 milligrams group continues to deepen down to -16.3, with a more modest decrease overall on average between the 2 doses. Next slide, please. Now, how does this compare with other phase 2 studies that we are aware of, who have progressed from phase 2 to phase 3? And here we took on average the placebo rate, which is around -6.3. But if you look across the active treatment, whether it's omalizumab, Remi, Rilza, or Bazo, the clinical activity range that you see here is from -9.1 all the way to -20. So clearly, we do achieve with the 60 milligrams group UAS7 reduction, with it, which is within the range of therapies that have moved into phase 3 and is differentiated from placebo. How does this compare with phase 3 data? In the next slide, please. We also see fairly similar placebo range, which is perhaps slightly more at -7, but a clinical range which is also interesting that starts from -8.5 all the way down to -19. Now, those of you may remember that dupilumab at week 4 do not achieve substantial reduction in UAS7, but takes just that little bit longer to get to the clinically impactful UAS7 later on at week 24. So in the next slide, what I hope to show you are the UAS7 reduction in some of the subpopulations that we've looked at. So on the left-hand side, you will see patients who have more severe CSU at baseline. So in other words, CSU patients who have UAS7 score of 28 and above. And here you do see that the 60 milligrams group have a much deeper reduction of UAS7 at -15.4, and also a little bit more with the 120 milligrams group at just under 9. In addition to that, we looked at a group of patients, albeit the numbers are very small. We have 3 patients who presented with angioedema at baseline, very severe angioedema at baseline. And here you can see that the reduction is around -18.7. In addition to these 2 subgroups, in the next slide, please, we also have patients who have low IgE at baseline. And those of you may remember that some of these patients are more indicative of the type 2B endotype, and they're more difficult to treat. Here, the UAS7 reduction is around -12, but bear in mind that the placebo rate taken from REMI and BARZO data, phase 2 data at week 12, placebo average is around -3.8. So it's a lot more differentiated here in this particular group of patients. Next slide, please. And this is disease control as measured by UCT7. I'm pleased to say that all 3 doses achieve a 4-point improvement or more in UCT score. And those of you may know that a UCT score that is minimally clinically impactful difference is 2 points and above. And here, both doses achieve more than 4 points. And on the right-hand side, you will see that UCT indicates at least 30% of patients achieve good disease control at week 4. So next slide, please. So in summary, I hope I've shown you that our 60 milligrams dose clearly achieved efficacy within the clinical activity range observed from phase 2 and phase 3 studies. And in some subpopulations, particularly those with more severe disease at baseline, those with angioedema and low IgE patients, the reductions are more substantial. INF904 also shows good improvement in disease control, and I'm also pleased to say that the drug was well tolerated and did not result in signals of safety concerns. So we are also grateful for the continual support that we're getting from Professor Martin Metz, successor to Marcus Maurer, and further exploration into potential CSU program will be discussed and provided at a future R&D event together with and also HS. So with that in mind, we would like to also now, we are thrilled to have with us Professor John Ingram, that I hope have joined us to start our Q&A session. And in order to do that, perhaps we can kick off by asking Professor John Ingram that in his view, in your view, as an expert and treating physician in HS, what is your impression of the data? And perhaps related to that first question, what do you believe matters to your patients and to you as prescribing physician when it comes to measuring progress and or improvement? So those are the 2 questions. I don't know whether John.
Yeah, we're still waiting for John. So what I can do is, if you could just hold that thought, Camilla,
okay,
run through some of the questions we've received, and and then address him when he gets on board. So just quickly, at this time we'll start the Q&A session as we wait for John Ingram. You may ask a question a few ways. One is you can raise your hand using the icon below the presentation window, and then we'll get people individually. You can also use the chat, the Q&A button below the presentation, and I'll read those questions out loud. So with that, we've got the first question from Steve Seedhouse at Cantor. Steve, if you could accept the invitation to unmute and go live, and you can ask your question.
Great. Yeah, thanks so much. Good morning. Congrats, obviously, on the really encouraging data here. I wanted to ask a few questions. First, I believe the study, according to the clinicaltrials.gov entry, enrolled across quite a lot of sites in the US and Europe. Just hoping you could comment on maybe the distribution of enrollment across those sites, particularly in the HS study. Start with that question.
Sure. Yeah. Niels, I think you could take that. I think you need to unmute perhaps, or no? No, you're good. Sorry.
Yeah, I'm unmuted. And Camilla, you try. I mean, if I say something wrong, but we had that's right. We had a bit below 30 study centers, so I think 28 actively recruiting in the two arms. Not every center recruited in both in each arm, obviously, but we had a good distribution. Like roughly 40% of the patients in the HS arm came from the US. And then I think the largest site recruited 4 patients. So there was no single center that recruited like the majority of patients or so. So very well distributed over maybe like a total of maybe in the range of 13 to 15 sites in HS and similar site like level in CSU. I'm assuming you were most interested in where HS was recruited, but happy to give you more color on CSU as well. Camilla, I don't know if you could speak to the site distribution in CSU.
Yeah, absolutely. So similar, we have our US sites as well as Western European sites coming from Bulgaria, Germany, Poland, Greece, and also Georgia.
Great. Thanks for that. Just mechanistically, also, Niels, I mean, nobody's been studying complement longer than you, and also HS. Curious if you could comment on the draining tunnel data, the pain data, and maybe also the DLQI data are sort of where the most profound effect you see is. Is that what you expect with the mechanism here? Like, how is that fitting into the sort of hypothesis of this mechanism in HS?
Yeah, absolutely. Great question. Thanks, Steve. So maybe noteworthy before I go into answering directly, that the C5a receptor expression has been found to be increased in various immune cells around all 3 types of lesions, abscess nodules and draining tunnels, and in all stages of the disease, like in early stage 1, 2, and 3. Now, early system is more like a chronicity kind of thing rather than a severity thing. But what I'm trying to say is the receptor expression is regulated in different cell types. Now, we've always been pronouncing the role of neutrophils in this disease as it's clustered into the so-called neutrophilic skin disease bucket. And obviously, they do play a big role, particularly in draining tunnel and abscess formation. And why is that? Because C5a induces netosis in these cells and the production of granular enzymes and also of oxidative radicals. So if you think about the impact of netosis has been implicated in the last few years more and more by the experts, the HS experts, to be driving, and also not just driving like draining tunnel, existence and pus formation, but also the actual development of draining tunnels. So the netosis angle is an interesting one, and we really cater to that. On the other hand, there are different other cells like immune cells, like monocytic cells, histiocytes that show a strong expression, and they also modulate the inflammatory environment. So when thinking into pain, if you change the inflammatory environment, you may work on like 2 different angles. I mean, there's certainly the pathogenic pain, if you will, like the destructive, like tissue destructive angle of neutrophils and other cells that will cause, certain dendritic cells or certain receptor, like just to be damaged and causing pain. And then there's also potential direct angle into the nociception of pain that has been described in the literature. But on a more kind of high level, if you do change the cellular component and the activation status of these cells in the skin, you actually, what you are expecting is not just to be active on draining tunnels or their formation, but to over time, particularly also move abscess and nodule counts further down, further down, further down, because you're kind of taken away one of the driving factors of the disease, which is really the immune cell driven inflammation in the skin. So that's why we are so encouraged. And we do believe it works on all three lesions. There's data in the appendix as well on the total inflammatory burden of disease, which is the ANDT count. And also there, a clearly and differentiated movement from any placebo ever reported. So I would say, the pain is the most impressive one because it's an integral part of the disease that is oftentimes not well addressed. But at the same time, I would not focus that we're just working on draining tunnels. I mean, we're really working on all three lesions. And that was the belief that drove us back into the disease. We all had made our experience, like we recently all made experience again through another big readout that high score can be very difficult because particularly controlling placebo responses. And we have a good idea now why that is. We've done a lot of modeling. So I paused here. I could go on and on, as you can see, but I hope I addressed part of it by really the immune cell composition in the skin.
Yeah, no, I appreciate that perspective, Niels. Just one last quick one for me. The IgE experienced arm 3 of the CSU trial, just an update on the status and the timing maybe of when you might have data from that. Thanks for taking the questions.
Yeah, absolutely. I will hand this over to Camilla. Camilla, do you want to take the CSU part?
Absolutely. Our arm 3 is challenging, as you know, with this particular patient group. So we would like to take a review by the end of November to see how far we have got with that. But those numbers are difficult to come by. We will announce the progress in due course.
So no clear guidance yet when we expect that to be fully closed out. But it's been proven very difficult. As you can probably appreciate, Steve, one of the challenges in these smaller trials is if you provide like a 4-week treatment outlook for patients with a chronic disease, it's not necessarily appealing for many of them. So it's not so easy to get patients on board. But we don't want to create excuses either. This arm is recruiting slower. And that's probably why it's usually just looked at retrospectively in the larger studies, but we're doing our best on that one. Yeah.
Thank you.
Great. Thanks, Steve. So we do have John Ingram in, but I do want to have one question for Camilla, a very important one, and I think in our case, a very easy and fast answer. So that's related to the use of antibiotics in the HS study. If you could give us a sense of how that was used.
Absolutely. I'd be happy to take that. So first of all, patients are not allowed to have systemic antibiotics during the course of the study. And I'm pleased to say that no patients actually took it. So therefore, we didn't have any patients who were on systemic antibiotics. We did allow any patients who were previously stabilized on tetracycline-like drugs to come into the study, but I'm once again pleased to announce that we didn't have any such patients either.
Great. And Camilla, if you want to address John and get our insight from him.
Absolutely. So Professor John Ingram, thank you very much for joining. And we are delighted to be able to have you here to perhaps give your perspective as an expert and treating clinician in HS. Your first impression of the data that we presented so far, and then also a leading on kind of next level of question is, what do you believe matters to your patients and to you as a prescribing physician when it comes to measuring progress and or improvement?
Thanks, Camilla, and it's a pleasure to join you. It's for me, I'm a clinical academic, so I have 2 clinics a week and I do HS surgery, and those key issues for our patients, I think the early data is addressing. we have, of course, these 3 lesion concepts around inflammatory nodule, abscess, and draining tunnel, and all 3 are important. Probably the lesion type that's been most difficult to treat has been draining tunnels, and that's where I think some of the additional focus is at the moment. And one of the issues there is that they can drain continuously for weeks or months. So an abscess might last 10 days and be acutely painful. It may give a pain score that's gone from 0 or 1 out of 10 to 10 out of 10 for that 10-day period, but then it will resolve. A draining tunnel will carry inflammation typically for a very long term, as I mentioned, months, quite routinely, and that produces drainage and odor and stains clothing and limits a person, and will be painful. So they'll have that pain continuously. So, I thought it's very encouraging to see the data that you have around within 4 weeks. I think we should be mindful that this is very short-term data, in that we have seen in treating our patients that if a targeted therapy is going to work, it often works quite swiftly. So this data probably does represent a demonstration of the mode of action being pretty effective in targeting HS inflammation. And that's not a surprise to us in HS. We've always felt that complement was an extremely good target for HS therapy. The innate immune system is such a big component of HS, and we've seen, as Niels has already mentioned, that those links with neutrophil biology, for example, where it's all part of that pathway, and interleukin-1 and so on. So this is very congruent with our thinking. We've been looking for a molecule that is effective against this target, and the early data is encouraging. And I think we look at the overall lesion counts, and they are greatly reduced, and I think importantly the draining tunnel count in particular, and that then is complemented by the patient-reported outcomes in terms of pain is the key symptom of HS. Is followed by drainage and fatigue. Those are the 3 actually that patients most value in terms of improvement. And then the overall concept of quality of life comes in. So that's the DLQI. You've seen here some substantial changes from baseline. The DLQI minimum important difference of 4 points is probably a bit low in HS, if to be very direct about this, that our patients need more than a 4-point decrease, but it's a pretty good start to at least attain that. And we can see here that, for example, on the high dose, we get a DLQI mean score reduction of 10 points. And those are the kind of figures that we really have been looking for in HS, because if you have a DLQI score of between 20 and 30, you're going to need it below 10 to really demonstrate that clinically meaningful change, as well as the minimal important difference. So the data is, of course, week 4. We would anticipate deepening of response going to week 12 or 16, primary endpoint of most phase 3 trials. But beyond that as well, although often it's observed case data, you'll see at least maintained, if not deepening of response beyond week 16 up to the sort of 1-year mark. So this is encouraging, very encouraging early data.
Great, thanks, Professor.
Thank you, John.
Ingram. So we do have a few other questions. Again, just a reminder that if you could limit to 1 question and 1 follow-up, that would help us very much. We also have now Ryan at RayJ. Ryan, if you could accept the invitation to unmute and ask your question.
Hi there. Congrats on the data. Question for me is the 120 mg dose looking like the go-forward dose, given the safety and efficacy data so far? And are you thinking about less frequent dosing cadences given the magnitude of the continued HiSCR50 response that you saw? I have a follow-up. Thanks.
Yeah, Niels, I think you can cover that, you and Camilla.
Yeah, happy to take that. Certainly, that's one route we are contemplating right now, just given that it's performing the best on every angle of the data set. But we are still doing some PK workup. We have not yet gotten all samples in, as you can see, and we will also do simulations that will further inform us. Now, to your second part of your question, yes, we do see potential ability to maybe move this to once daily dosing, but maybe not right away. We're trying to assess this right now because we've seen, as you know, like when we take patients off drug after we've loaded them for full 4 weeks, that we saw quite a good continued response after we taken them off drug. And the preliminary levels we see at week 8, 4 weeks after stopping of dosing, are encouraging. So maybe we can, after full loading, like also look into once daily dosing. So that's something that will come out of the simulation work. So we can't fully answer it, but certainly we're not currently planning to go higher than 120 mg. That's clear. But we're also trying to assess the abilities in between 60 and 120 for sure.
Got it. And then regarding potential prioritization, how are you thinking about prioritizing upcoming clinical studies and clinical progression for HS and CSU based on this data set? And do you anticipate a meaningful target dosing difference between these two indications at this point? Thanks.
Yeah, very good question as well. I just take it in the interest of time. Certainly, we judged both data sets as warranting to forward the development. Our key interest is in probably also a market that is very interesting just from the medical need, which is HS, and I think the data clearly warrant that. So there will be an initial focus here to move forward with HS. But that is not to say that we've looked at the CSU data a lot and we feel there's a clear differentiation to placebo. Obviously, there is the ability to move forward into a deeper response with also targeting not just the histamine/mast cell angle, but also the angle in like tissue inflammation, which is going on in that disease as well. So there is this clear wish to further progress it. And we think this is a pipeline and a product potential that we have in our hands. So while we focus maybe initially to drive start in HS, there's clear, I would say, support internally and also clear wish to move it forward in CSU, but also beyond in some other indications. We mentioned previously the renal space, peripheral neurological space. And as you probably appreciate, there's renewed interest here in the area through a recent deal, but also through a marketed drug that's bringing real sales. So from that angle, we clearly see this potential. Thank you.
Great, thank you.
Thanks, Niels. And then for those that are queued up, we do offer Professor John Ingram to answer your questions in HS if that's of interest to you. So so Camilla, this one's for you. It's regarding the data we saw in the CSU arms. I'm wondering if you can give us your thinking on what we might be seeing there in the context of other successful drugs and target coverage and things like that.
So I think what's encouraging about CSU data is that the 60 milligrams group, a bit like in HS as well, you see efficacy with 60 milligrams. We cannot fully explain why we see the modest decrease with the 120 milligrams in CSU. We do know that that there are a couple of patients who seem to skew the results because of flare in one particular patient in particular. But, I guess I like to bring your attention back to the development in omalizumab, right? The 600 milligrams group actually ended up having worse UAS7 reduction than the 300 milligrams. Now, it's early days, and as Niels mentioned, we will be doing further PK analysis as well as PK simulation to really try and understand the efficacy that we should be focusing on. But I think we come away being very encouraged that 60 milligrams show good efficacy.
But if I may add, Camilla, and also to the earlier question of Ryan, the 60 milligram group, particularly also in CSU, provides for it's preliminary PK data, but it provides for a real good and high exposure as well. So from that end, it's just to to basically reiterate what you just said, that promotes that 60 milligram is a viable way forward too, right? And you just basically also said, in other words, we don't fully know yet whether there's an inverse dose response here, or whether that is just a matter of sometimes in small trials, you may have a few patients having a large impact. And when you look at larger data sets, that may not be the case anymore. But yeah, that being said, like 60 is viable from an early PK look, right?
Exactly.
Great. We've got one question from Catherine Della Russo from LifeSci. Catherine, please accept the unmute request. Catherine, I think you might be live. Go ahead, try. Okay, so we'll go next to Andreas at OPCO. Andreas, if you could accept the invite.
All right, great. Thanks, guys. A couple of technical difficulties here. Anyway, congrats on these results. Two questions for us. Thinking about the phase 2b in HS, would you be interested in including an active comparator in that study, and maybe some of the additional comments around potential dosing arms there, duration, primary endpoint? I know you showed a couple of very interesting data points there. And then just in CSU, you saw consistent responses across low IgE, high IgE patients, and also strong effects in severe and angioedema subsets. So do you plan on kind of enriching the phase 2b/3 study for a subset of those patients? And could you use a baseline IgE as a predictive biomarker? That's for us. Thanks.
Okay, maybe I jump in first and then Camilla can jump for. Thanks, Andreas. Lots of really important questions, clearly. I think, for the HS part, we haven't yet fully done the simulations on PK, but we anticipate maybe at least 2, maybe 3 doses moving forward with placebo. Your question was, do you want to put in, or do we plan to put in an active comparator? I know that it's been, I think once or twice been done before, but if we do so, we haven't fully excluded this possibility. What I don't necessarily like, if you have a certain power assumption, and if you like to do a certain study, just to put a smaller arm in, because I want to come back, a lot of people judge like efficacy on high score, and in small data sets that can be very deceiving, right? The problem with that is, again, if you have 2 data sets that look in mean baseline and end count the same, they can be very differently distributed in terms of, the question is how many low end count patients you have. When I say low, is maybe below 10 in the placebo arm. And if you by chance are in that noise, those types of things can skew results. And so if you have 2 non-comparable data sets, that's something we would want to avoid. So if we did put in, and it's not finally excluded that we will put in a comparator, it's just maybe less likely in my eyes, but it's possible. We'll also do work with experts to discuss that. We would then put it in at the same size and not as a small arm, if that makes sense. And so to the other questions, I think in CSU, I'll definitely hand over to Camilla here in a second. But yes, I mean, we are very encouraged by some of the findings in the more severe subgroup. Why? Because when we discussed this with our expert, Professor Metz at the Charité, he mentioned this angle that I mentioned before. If you're not just treating histamine from mast cells, and clearly the receptor is expressed on mast cells, but if you are also changing the environment, that may have a different impact, longer lasting. And he reiterated 2 things. The one thing is, if you treat that tissue inflammation, that may be particularly important for those that have a very inflammatory phenotype. And, talking about angioedema, if you think about how many patients have angioedema history, it's roughly 40% according to literature. So that it is a sizable subgroup that could be of interest. but we haven't made that decision yet. But I hand over to Camilla to give more color or her thoughts on moving forward in CSU.
Yeah, sure. I mean, currently lots of great ideas coming our way, and also in discussion with Professor Martin Metz. I mean, I think we would look at the group with so-called, who have more inflammation, and those who are indicative of, type 2B endotype, low IgE. But I would say, we still have ARM3 ongoing, and I think results from that would also help inform in our planning for phase 2B. So I think we could potentially enrich, but enriching this group with low IgE will also take longer, and that's been our experience. So it's possible, but also in terms of timing, it will take potentially longer. Using baseline IgE as a potential biomarker, I mean, I think baseline IgE is informative, but I'm not sure if it's a perfect biomarker, because as you know, if we wanted to go fully for type 2B endotype CSU population, we also need to think about potential other tests like, basophil stimulation tests, BAS and BRAF, and so on. These are not commonly done in a lot of hospitals. So, it's possible that we may want to think about sub-studies to really see where else we can enrich, right? And how else we can enrich the population. So all things are possible, and it's still ongoing. So hopefully more to come at a future R&D day. Thank you.
Okay, great. Thanks for taking my questions, and I'll hop back in the queue for someone else. Thanks, guys. Congrats again.
Chris. We do have a question
you,
in the chat. Niels, this is for you, related to some interest, right, in the C5aR mechanism in recent weeks, and given where we are with the product profile, where we see potential avenues of further development outside of HS and CSU. Perhaps some low-hanging fruit on that.
Yeah, great question. So first of all, I want to reiterate, I think these data further support that we may have a best-in-class asset here in our hands, just upfront. The C5aR as a target is really well researched. There's a lot of studies, C5a, C5aR, over 6,000 publications in PubMed. And the target is really indicated in many inflammatory diseases, particularly many autoimmune and autoinflammatory diseases. There have been, over the last, couple of decades, lots of efforts to drug this target, but it's a tricky target. C5a, we know really well, is a tricky target, but C5aR is also not easy, because that's the big contribution back then from Chemocentrics, who got acquired by Amgen for $3.7 billion some years ago, is they found that allosteric binding site inside the pocket of the receptor, that when you target that and your drug gets in there, then you can really evoke a complete blockade of the signal. So that discovery was important. The cyclic peptides in previous history, they all had toxic side effects. They were good blockers, but toxic. And so the chemical ones, it was difficult to target, as I mentioned. However, targeting that allosteric binding site means your drug is very lipophilic to get in there, and then becomes a formulation issue. And that's really where we worked several years on. Can we find an angle to get to that same allosteric binding site, but with a formulation and a drug that has a better PK, and therefore blocks that much better in the human system, in the in vivo system. And that's where our drug is differentiated. So recent activities, there was a recent Biogen, I think, agreement with a small company that's very early. We don't know much about that product. That's been about, I think it's a couple of years away from even IND filing, so we can't really say too much about it, but I think that further indicates there's interest. So long story short, target is well known. If you can drug it and you have a drug that fulfills all requirements, I think it's a very interesting pipeline and a product potential. And I come back to my first sentence. I think we now have a best-in-class target drug in our hands, and we're really excited to move it forward.
Great, thanks, Niels. Catherine, if you can hear me, we'll try you again. I think your mic should be working.
Yes, thank you, and I apologize for the technical Here, this is Kate on for Sam Slutsky. I'll be quick here. Did you happen to measure C5a levels in the studies? And was there any correlation between patients who responded better to those with higher C5a baseline levels?
Catherine, really good question. I just jump in. We don't have this data yet. We have, though, in previous times not seen that the baseline C5a levels, which are elevated in HS, for example, clearly, and also have been reported to be elevated in CSU, they're not necessarily a good patient-by-patient like indicator whether your drug works or not. And that has to do with a lot of intraday variability about how much C5a is produced, but also with a simple fact: if you have very high C5a levels, you know there's complement activation going on. There's no other way. But if you have low or not elevated C5a levels, that can be just because you can't exclude that complement activation is going on, because there's so many receptors around that you may just not see the elevation yet because they're soaked up by the so-called big sink. So our historic experience is that C5a levels are not necessarily a good initial guidance. Obviously, this drug, just reiterating, it doesn't block C5a, it goes to the receptor, so it wouldn't be a PD marker for our drug either. But we don't have the data in yet. We do measure C5a levels, and we can probably say more at a later time point.
Great, thanks so much.
Sure, thank you.
Thanks, Kate. Also, we now have Will at Leerink. Will, you can ask your question.
Great. Hey guys, thanks for taking my question and congrats on the data today. Really great to see. So just one question on the pain data for NRS 30. It seems like there's a flattening of response between week 3 and 4, but then on high score and some of the other endpoints, even draining tunnels, we see a really nice improvement between week 3 and 4 still. So I guess just curious what you make of this dynamic, and is there anything here that we should appreciate as we dig into these data a bit more? And then I have a quick follow-up for Professor Ingram. Thanks.
Yeah, absolutely. Good question. I mean, for the 75 milligram group, it looks like this, but I want to reiterate 75% NRS 30 and a 2-point reduction. No one has delivered that at week 12 or 16 ever. So maybe it is real that we don't see much more than we see already between week 3 and 4. So we will get more granularity on week 8 data to make that point. We don't have that yet. But if you look at the dose groups below the 120 mg, they seem to be still moving. So is the 75% NRS 30 response the highest we can get to? I cannot answer that yet. But if it would still be, at least to me, pretty impressive and very differentiated from the rest. So sorry, I can't give you more at this point in time, but we may have more over time. Yeah, I think you also had a question for Professor Ingram.
Yeah, great. Thank you so much for that, Niels. Appreciate it. And yeah, just for you, Professor Ingram, appreciate your time today. I guess just quickly thinking about the evolving treatment paradigm in HS, it's seen a lot of recent data sets over the past 12 months or so. But when we think about INF, I mean, I could see it being a very valuable second line option for people who, failed biologic therapy, but then with this really rapid onset of action here, it seems like it could be a very valuable first line option as well. So just curious to get your preliminary thoughts on how it might fit into the treatment landscape, assuming these data are replicated in future studies. Thank you so much.
Hey, Will. Yeah, I think I was also going to comment on your pain question as well, because some of that flattening until week 3 to 4 may in part reflect that you have to attain that at least 3-point reduction in pain. And when baseline means are around 6 or so, there may be that, to some extent you kind of produce that rapid improvement. Then there's a bit of residual pain, for example, from chronic scarring as well in HS. So maybe that some patients, it's quite hard to get their pain scores down, to complete sort of low levels because of that scarring too. But in terms of where INF might sit in terms of the treatment pathway in HS, some of this will depend on the delta really, in terms of comparison with, for example, anti-TNF biosimilars. The payer will be mindful of that. If there's a demonstration of improved efficacy beyond those biosimilars, then the cost-benefit equation may allow to be the first targeted therapy. what we've seen, of course, in recent times is that phase 3 results, there's nothing that's a substantial improvement above, say, adalimumab biosimilar or adalimumab in the Pioneer studies in terms of its HiSCR 50 attainment. And so there's probably then payers might consider this, subsequent target therapy to be second line after the biosimilar on cost grounds. What we look for now is for a targeted therapy that performs substantially above those levels so that we can advocate that they should be first line as a targeted therapy. And that's where I think there's an opportunity, because HiSCR 50 is not a very high bar. Someone might have 20 inflammatory lesions. To have that reduced down to, say, 9 or 8 is not remission. It's not even minimal disease activity. It's still ongoing active disease. So we're now looking to see if we can get high efficacy bars and get increased attainment, and then we could ideally look for those new therapies to be positioned potentially even first line in the targeted therapy pathway.
Excellent. I really appreciate the insights there, and congrats again, guys. Thank you.
Thanks. All right,
Thank
folks,
you, Will.
I think we'll have a couple more questions. I know we're a little bit over time, but trying to cover as much as we can. Our next question is from Dev Prasad at Lucid.
Hey guys, congrats on the data, and just one question from me. So now you have dosed several patients with INF904. What's the emerging safety narrative for regulator, and are you any class related concern you are proactively monitoring? Thank you for taking the question.
Absolutely. I may take a first step here. So, yeah, you're right. At this point in time, we have no safety concerns that emerged from all that. And remember, we tested very high doses, up to 240 mg in the phase 1 study. So I think from that end, we're good. We have a preclinical package up to 9 months, non-human GLP, non-human primate tox study, and also no safety signals of concern and no real dose-limiting toxicity detected. And so from that angle, we see an interesting opportunity for a safe oral in the future. Obviously, we have to still prove it, but so far it looks like that. Now, what are we monitoring? There's no class effect known, so I think generally blocking the pathway it can be considered very safe. There was one liver-induced toxicity in the avacopan approvals, and there have been some signals since, but that's related to avacopan in our eyes. It's a very decent inhibitor of the CYP3A4/5 liver detoxifying machine, as I call it. And we have shown preclinically that we are 36-fold less engaging with that enzymatic apparatus. So very likely we don't have that problem, which would be great. Are we monitoring anything? We are always monitoring safety, obviously. And one thing for any immunomodulator is always, generally speaking, if you have like an infection risk, for example, over time, and that's also expected by regulators to monitor that. Now that will be only looked at in like controlled studies that are longer term dosing. But really, yeah, from that angle, I think that covers the question. I don't know, Camilla, if you had additional thoughts on that one.
No, you've covered it. Thank you.
Okay, thank you.
Great. So one more question, I think, Niels, and I think Professor Ingram, I think a good question for you regarding just thinking about potential synergies with other mechanisms in HS, for instance, IL-17s. Clearly, it's early days, but we like to speculate on things like that. So, Niels, I didn't know if you had any idea, or Professor Ingram, any thinking?
I would really, really defer here to Professor Ingram first line. John, I think you're much more position to really. I have my biological ideas about that, but I think you, as a treating physician with so much expertise in HS, I think I'd like to kick that question over to you.
No problem, Niels. Yeah, and I think it's a great question, because we've seen so far that monotherapy is almost a luxury in HS, that we've yet to really see a single agent provide us with a universal minimal disease activity state in our patient cohort. So we're already using co-treatments routinely in terms of a targeted therapy plus, say, an antibiotic or an anti-androgen therapy. And certainly, I'm aware of case reports and colleagues who are using two targeted therapy agents to manage their most challenging patients with the highest inflammatory burden. So it's very much the case that we have these multiple pathways in the HS pathophysiology, the inflammatory soup that exists in the dermis of HS lesions. And so, of course, there'll be safety considerations, there'll be cost considerations when we combine more than one targeted therapy. But in terms of reaching those higher efficacy bars, that's certainly a possibility that is already in use. And we're also familiar with bispecific options and so on. So I think that it's a compelling option, but I think, of course, it's quite a fair way down the road in terms of current development of INF and others.
Thank you, Professor Ingram. So one more question, Niels. This one is for you since you punted on this last one. It's regarding just giving a little more color on what you think we're seeing with the PK data and what we're thinking about the end of study portion where we saw continued efficacy in many patients even 4 weeks after dosing has stopped.
Yes. So maybe I start with, I mentioned that we have levels detected. That's preliminary, so we don't have all the samples in, but the preliminary one suggesting a range of plasma presence in the range of, let's say, 50 to 200 nanomolar, and that's about where that's the range to where avacopan accumulates at week 13. So that's why we're saying we still have drug levels that are able to block. How much I couldn't tell, and that depends patient by patient. We also want to see if this is more pronounced in the 120 group versus the others. We have to still see this data to be more granular and get more data in. Our IC50 of the drug is around 25 nanomolar, which is pretty much exactly the same like avacopan, but our ability to reach a blocking level in plasma and in human tissue is clearly higher with our differentiated PK and formulation. So that also illustrates that these levels are over the IC50. So from that angle, we are confident that what we see still provides some blocking. Now, clearly not the same level as during full dosing. And the idea is now to do simulation work to understand what may the half-life look after we loaded the tissue after week 4. Can that be simulated? Can we, how long will we have full coverage of those 4 weeks? And maybe if after loading is a less high coverage enough to continue an improvement, right? These are the questions we right now have. We can't address them all with PK simulations, but maybe some of them. But I hope I could give you a bit more color on why we believe there are still active drug levels at the end. Yeah.
Great, thanks. So we are out of time. Niels and Camilla, obviously, thanks for joining us. Professor Ingram, thanks for joining us as well. For those listening in, you know where to find us. You can reach out to us if you have any questions or need any follow-ups. We are available via email. So again, thanks to everybody. You are free to drop off.
Thank you.
Thank you.
Bye-bye. Hey.
Anyways, I think there are still 90 participants. I'm not sure if we're off or not.
Yeah, I was wondering that too. Anyways, for everyone who's still on, I wanted to say big thanks for all the contributions, all the work, particularly for John Ingram. I know he had clinics this morning, so he was hard pressed to get in. and he's been very patient with us. So thanks so much, everyone, and the entire team for making this happen. MC Service team, you did a great job. I think this went all smoothly. And yeah, I hope we could cover some analyst question. Obviously, there's only that much time, but hopefully we can have discussions now with them after the release here. All right, big thanks to everyone. I will jump off as well in case you still hear me. Hopefully you got the thanks, and we'll be in touch shortly. Thank you. Bye.